Okay, I believe it's time. So it's my pleasure for today to introduce Alexia Cosby. Alexia graduated with a Bachelor of Science in Chemistry in 2017 from the University of Oregon. And then she pursued a chemistry PhD at Stony Brook University, and her dissertation focused on utilizing luminescent lanthanides towards designing biomedical cancer imaging agents using Cherenkov radiation. I didn't know you could use Cherenkov radiation outside of a reactor. And in November 2021, Alexia started postdoctoral research in actinide chemistry at the Lawrence Berkeley National Lab. Her research is focused on working with actinides in the context of in vivo decorporation and developing theranostic
Radiotheranomics,
models with zirconium-89 and thorium-227. Welcome, Alexia.
Thank you. Thank you, everyone. This is honestly such an opportunity for me. I mean, as an early career scientist, just being able to talk about science is so important, and being able to communicate science is important. And I'm someone who comes from, I'm very proud to come from a very multidisciplinary chemistry background. I feel like I've done a little bit of a lot of different things. And that's actually what motivates me in my research and how I want to push my research forward. So coming and doing a postdoc with Rebecca Abergel at LBNL has been a perfect fit to just expand this like toolbox of tools to try to really dive into my main focus and my main interest, which is radiochemistry, designing things to go from the bench to the clinic. And so how do we think about that as a whole, and especially in the context of using a mouse model? So we don't want to use, obviously it's of interest to minimize how much animals we're using in this type of research. But if you think about, we do want to design radiotherapeutics, and we do want to design targeted cancer agents. So how do we safely do that? And how do we safely do that each step of the way? How do we think about economically producing from the bench, literally economically, all the way to the clinic? So to kind of visualize that, I like to think of it as this cyclic process where we start from studying the radioactive fundamentals. How does radiation work, and how do we want to try to utilize it for our application of interest? So then you think about the chemistry design. How can we design chelators or small molecules to harness these radioisotopes in order to actually utilize the properties effectively? This goes into then, for pre-medical applications, this in vivo mouse model, or commonly it's a mouse model, but you can also use rats. There's some other things. And then that leads into clinical research or FDA approval, which then hopefully spreads global treatment and awareness. So it's a whole process, and it really does come down to the very fundamental things that we do every day, that we try to study every day. At LBNL, we actually have a huge resource, which is being able to work with a lot of different types of isotopes. So when you think about the different types of radioactivity and the types of isotopes that you're looking at, you need to think about the application. And when we think about the range of isotopes that we have to work with, we have short half-life isotopes such as gallium-68 or zirconium-89, minutes, hours, it kind of depends on what sort of context you're looking at. There's also very, very long half-life radionuclides such as radium-233, americium-241. So I'm kind of putting this in a range of context for the half-life. We can do radiotheraноstics, which I'll define for you in a second, and we can also do decorporation work. And so actually is what makes LBNL resources and facilities pretty unique. So decorporation, what do I mean by that? Decorporation is where you say you have an instance where you are contaminated with radioactivity. Then we want to be able to establish a treatment program so that you can monitor clearance of that radioactivity. So very basic, that's the fundamental definition of decorporation. Then when we think about radiotheranostics, this is a multi-definition word. Theranostics, therapy, and diagnostic, so for diagnosis. So the idea is that you can have one mouse model, and you can switch between using a positron emitter for imaging and a alpha or beta emitter for therapy. So I'm going to talk about both of these types of projects, but I'm going to give you an outline of the context of the questions we are specifically asking. I've umbrellaed both radiotheranostics and decorporation as general types of projects you can work on, but what are we really looking at LBNL? One of the things that we're looking at with our decorporation studies is we're trying to think about how do you effectively chelate these radiocontaminants in vivo? How can we actually go about treating them? And specifically, what we're looking at is what happens if you inhale radioactivity. So if you think about this, you can't necessarily have a human model for these kinds of studies. Who's raising their hand to go sign up to inhale a bunch of radioactivity? That's not how these kinds of studies can work. And this is why we have to use a mouse model, because this can mimic what we can predict for the human outcome. So our questions related to theranostics is what is unique about our system, which I will lay out for you. And then what constitutes a good targeted alpha therapy system, which I will also go into. So to first talk about decorporation, we're really specifically thinking about decorporation of radiocontaminants from the lungs. So let's define this a little bit. Radiocontaminants are, I like to consider as radioactive particles or unstable elements that are released in an unshielded environment, as in you don't want it. You're not asking for any radioisotope. It's a contamination. So decorporation is the idea of capturing that radiocontaminant and releasing it safely from the body. There's a lot of interest in this. We don't necessarily have control over some kind of incident that might happen. So we want to be thinking critically about how do you treat either before a contamination event or how do you treat after a contamination event. So if you think about this very simplistically, a radioactive isotope is emitted in the environment. You then inhale it, or it's uptaken into your system. It distributes and then accumulates. Well, radioactive isotopes, almost all of them, they want to go directly to your bone. So they're going to just start depositing in your bone and accumulating forever. But what if we could give you a treatment source such that as you administer the treatment or the chelator, it binds the isotope in your body, and then safely you can then excrete it out via excretion pathways. So the idea here, questions that we have are, how can we effectively chelate inhaled radiocontaminants in vivo? Other people have maybe looked at intravenous injections or injections through other routes, but what happens specifically when you inhale? And then how are they excreted? And can you pre-treat or can you post-treat? So the chelators, I've talked a lot about treatment and chelation, but what do I really mean by that? What we're looking at are two types of chelators. One is DTPA, and this is actually FDA approved for decontamination, so it's kind of our gold standard. And we're also looking at the isotope americium-241. So americium-241 has a half-life of about 300 years. You don't want that just remaining in your body for any reason. So the other chelator that we're looking at is HOPO, which actually has a really high affinity for these transuranic elements. So we're going to be comparing how DTPA looks with the excretion pathways compared to HOPO. In addition to looking at these two different treatment regimes, two different chelators, we're also looking at two different types of treatment. I briefly mentioned before, one is the idea of prophylactic treatment. That's where before any contamination event happens, you pre-treat your patient. And the idea behind this is, what if there are military personnel that are going out that you want to pre-treat just in case for any kind of event? So that's one idea of something that we're looking at. The other idea is post-treatment. The fact that, or considering that you might be contaminated with some kind of radionuclide, and then you can treat, and then we can monitor. So I'm not going to show any actual mouse images, that's not actually what we do, but I do want to kind of discuss the process of these experiments so that you can kind of contextualize what goes into doing this sort of work. So first you formulate your isotope. We put the americium-241 in a buffer, and then you install the radioactivity solution. And I'm going to pause here and take a moment to show you where the trachea is. The trachea is right here. So when we install, we're not just going down the throat, we're actually aiming for the trachea so that the solution can be installed into the lungs. This has actually taken us a lot of work to perfect. It's a very difficult thing to do. So I'm just going to acknowledge that it's difficult. After you install the solution, you then treat with your chelator or you pre-treat. And then you wait for your chelator to circulate, usually about 6 to 9 days is the time period that we wait. And then, as the mouse is excreting and doing its processes, we collect each of these elements, and then we analyze how much americium is in each organ compared to the bones, compared to how much is being excreted in the urine and feces. So you can imagine that what you want is your chelator to capture all of the radiocontaminant and then just be excreted through the urine. That's what ideally, or through the feces. That's ideally what you would want. You don't want the activity remaining in your body. So here I'm going to show you some data. And when we talk about this type of data, we talk about uptake. So I'm going to present it as the percent injected dose per gram. That means that the percent of how much we put into the mouse divided by the mass of the organ. And we use the weight of the organ to normalize our uptake because every organ is different, even mouse to mouse. One liver might weigh different than the other. So here, what you're just looking at is the percent injected dose per gram if no treatment was given, so they've just inhaled the radioactivity. And I'm specifically just looking at the lungs and femur here, because we're trying to think about how the radioactivity is being eliminated from the lungs, and we also don't want to see uptake into the femur. So when you compare to the DTPA ligand, a little cut off here, we have a few different treatment hours. So pre-treatment, 24 hours, pre-treatment 2 hours, and then half an hour post-treatment. And so that's all relative to when we administer the radioactivity. And so here you can see that it looks like when you treat at about half an hour, that eliminates the most of the radioactivity from either the lungs or the femur or this ligand. But then when you compare to HOPO, we see even less content in both of those organs. So what this is telling us is that the HOPO ligand is doing a better job at chelating the americium from the body and excreting it from these organs. And I have some numbers here: 180-fold recovery from the femur and 34-fold improved equilibration from the lungs. Excreta analysis is another way that we analyze where the radioactivity is going in the body. So we can look at here, what I'm showing is the DTPA here in the urine. So this is the americium content in the urine. So what you would want is high amounts of americium in the urine or feces. Unlike in the organs, you don't want high amounts of americium accumulation in organs, you want it in the feces. And so here we're showing that in the urine, the DTPA clears it out much more quickly compared to the feces. And what you actually see for the HOPO is a different trend, where the HOPO ligand passes through a different pathway. So it's actually excreted through the feces more than the DTPA. And so this is exactly what we've seen with other ligands in past literature. So everything is making sense in the context of what we've tried before. And it's really interesting that the treatment regimes are collaborating with what we've seen in terms of uptake in the organs. So, okay, we can do it. We can treat. It looks good. Another question that we had is, are we actually installing the right amount of radioactivity? We can't see where it's going, right, when we're doing the actual procedure. But what we can do is we can switch out the isotope that we're using. So rather than americium-241, which decays by alpha emission, what if we swapped out and used zirconium-89, which decays by beta emission, which you can use for PET imaging? So what we did was we took a mouse and injected it with a little bit of zirconium-89, and then we did some imaging. So this is a PET image of a mouse. Coronal means that the head is up here and the tail is down here, and you're looking at it just kind of flat on the bed like that. And then sagittal is side on, so you're seeing the side. And so here, 30 minutes post-inflation, you can just see it's not a lot of much. You don't see much, but it's uptake through the lungs, and that's the signal that we're seeing. And it looks even better as an image or as a video, but I don't have that. And then after 24 hours post-inhalation, what you're seeing is accumulation and uptake into the spine, which is exactly what we would predict for zirconium. So this is just kind of a proof of concept that we can visualize that we're actually installing the correct amount of material into our lungs. So just to kind of summarize and wrap up the decorporation aspect of what I'm talking about, we can visualize it using PET, and then we can actually see an improved decorporation from the lungs using the HOPO chelator versus the FDA-approved DTPA, which means that this HOPO chelator could push forward and use for much more things. So with that, I'm actually going to transition now and talk about radiotheranostics. When I first showed you all of the in vivo work and mouse work that we can try to do at LBNL, yeah.
This is really interesting and
Yeah.
really good. Just a question here on the error bars. I mean, it looks actually like, I mean, it's hard to tell because it extends below.
Yeah.
Is it statistically significantly different when we look at the aggregate sum on all time intervals between DTPA and HOPO? It
Yeah.
looks like it's a big error bar.
It definitely is, and we're still actually performing statistical analysis on each of these, but we do have full organ data as well, not just for femur.
Right.
But you're right.
If you put all the organs together, maybe look at the whole thing, you could see
Yes.
that a small error.
Yeah.
Okay.
Yeah. And I
And-
mean, it brings up a good point. Data analysis is really important to this type of error analysis is important to this type of project. So.
And one other question,
Yeah.
you showed the clearance of the urine and feces there. Were the error bars just so small we couldn't see them on the data points?
Those are
Because it looked like a much bigger number.
yeah, unfortunately, the way the actual experiment works is that is a value for 4 mice total altogether. So those are individual values that have been summed.
Got it, yeah,
Not
so your error bar
masses.
gets smaller.
Yep,
Sure.
yep.
Thanks.
Are there any other questions? Yeah,
So I was just wondering, like why you chose half an hour specifically for the post-treatment?
I think this was based on kind of clearance pathways that we've seen before, not for intratracheal, but for IV and IP installation of the radioactivity and the treatment solutions. And so we're just kind of looking at really close time points. So half an hour was also within what we could do experimentally. Yeah. Yes.
Do you remove the metal from the bone, or once it's uptaken from the bone, it's pretty much just gonna stay there?
It's a good question. I'm pretty sure it's just gonna stay there. It's pretty difficult to, but like the bone structure, hydroxyapatite, those groups are just really difficult to try to pull back out. Yeah.
This is really cool. One question I had was, what sort of uncertainties do you have to consider when you're using zirconium as a surrogate for the imaging versus the actual decorporation or therapeutic isotope? Like what differences are there between like the distribution of one versus the other?
Yeah, that's a really good question, especially for what I showed with zirconium. It's a 4+ metal. Americium is a 3+ metal. They have different decay routes. They have different daughters. The zirconium decays to yttrium naturally. So those are considerations. What I haven't shown is any kind of PET imaging with treatment. And so you actually want to consider the binding of which metal that you're choosing. So HOPO binds americium pretty well, but does it bind zirconium as well? Of which it should, but you want to think about those types of, those are all really good, questions that you're thinking about. In this case, zirconium is just such a commonly used PET isotope that it's a pretty easy and convenient to get your hands on and monitor biodistribution. Yeah. Anything else?
And I assume that when you've got an alpha emitter, everything is going to be different anyway, right?
Yeah.
Because you have recoil, which you don't.
Yeah.
With the PET.
That's true too. Yeah. Yes.
When the zirconium was put into the mice, was it already related to something else, or?
No, it was in a buffered solution, a citrate buffered solution. Yeah, yeah, okay, I will move on into radiotheranostics. Basically decorporation, I like to think of if we're trying to think about chelation and chelation therapy and treatment, those are really, really long-lived isotopes. If we think about on the shorter scale, you can actually utilize a bunch of different properties from the periodic table. There's a lot of different isotopes there. It's referred to in my field as a nuclear chocolate box, if you will. So radiotheranostics, we like to think of it as streamlining personalized medicine through isotope exchange. And what do I mean by that? This is one of my favorite images from this paper here. And it basically works by going through 3 phases, if we want to think about it like that. First, you administer your diagnostic phase imaging agent. So for example, zirconium-89. Zirconium-89 is a imaging isotope, but it is not a therapy isotope. So first you administer the imaging isotope, and then you go through the therapy phase once you identify, okay, there's the tumors. Then you switch out your radiometal and give the therapeutic phase. And then you can follow up and continue imaging. And ideally, it's just the same metal that you're switching out. But there's a lot of different considerations into developing these types of systems. So I'm going to give you an FDA-approved example. And I know it's a big organic structure up here, so I'll break it down a little bit for you. There's the radioisotope up there, and it's chelated by DOTA. This is a very common chelator used in radiotheranostics. And then this whole thing right here is just our targeting peptide. So that peptide is a well-established targeting vector. We know it's going to go target the somatostatin receptors. So we basically just pick, a well-known targeting vector and then append our radionuclide. So how does this work in the clinic? First, you label with gallium-68, which is a positron emitter, and you administer it to the patient and you image. And so this is pre-treatment, and you can see all of these lesions here, all of those dark spots. This is a CT image. So then you administer lutetium dotatate, and this is what's FDA approved. It's labeled with lutetium-177, which decays via beta emission. So it's not an imaging radionuclide, it's therapy radionuclide. And then it has a half-life of 6.5 days. So you administer the treatment, and then you follow up with another PET scan or PET CT scan, and you can see the dramatic improvement of the decrease of the lesions after the treatment with the lutetium-177. So I really like showing these images because it gives context to, we have one targeting peptide, we have one chelator, and all we're doing is swapping out the metal. But of course, it's a little bit more complicated than that, right? What are some of the issues that are limiting this theranostic development? Why are we not all very familiar with getting a theranostic radioisotope injected into us? And why is there kind of a paradigm of that? So what are some issues? A big issue is that those big organic things are difficult to make. They take multiple steps. When you take an OChem lab, multiple step synthesis, and often these are low yielding. They're very difficult to make. And having something, if you really want to push it into the clinic, needs to be a little bit easier to make. So that's a huge limiting factor. We have a demonstration here. I've shown you that HOPO chelator. Here's that same HOPO chelator, the zirconium-89, attached via some spinach to a targeting molecule. And here they've shown, here's some PET images. They've shown uptake into a tumor. Yay, that's great, it works, but it takes 13 steps to make this. No one's gonna wanna go and work really hard to make this ridiculous compound. Can we try something easier? Is there some way to basically push these things together more effectively? One thing that we have in our toolbox that we've done a lot of work with at LBNL is this protein. So this protein, siderocalin, naturally acts as this antibacterial agent by sequestering siderophores and their iron complexes. So I've shown you that HOPO chelator. This protein is kind of a chelator of a chelator. So enterobactin or iron enterobactin, same with HOPO, it'll chelate iron naturally from the environment, and then the siderocalin protein will gobble it up and recognize it, and it stays firmly sat in there inside the pocket. So we've also established done a lot of work with siderocalin binding both lanthanide and actinide complexes. We can get these really nice crystal structures showing that the HOPO complex of americium mercurium fits really nicely inside that pocket. And we can also use this to kind of elucidate some other interesting bonding properties of F elements, because we can crystallize these and look at the protein structure versus the small molecule structure of these compounds. But what I want to emphasize to you is the high binding affinity of these complexes inside the protein binding pocket. We've established that this siderophore protein loves these complexes. It's going to grab onto it and it's not going to let it go. So we're thinking, could we use this to our advantage for a theranostic? Could we use the fact that this is a squishy protein to try to develop a targeted theranostic agent? So how this would work is through a fusion protein, and the actual fusing of these proteins was not done by us. It was done by our collaborators, Roland Strong, at the Fred Hutch Cancer Research Center. So I'm showing you here a structure of the siderocalin protein, and what I'm calling, it's a different ligand now, just work with me, a ligand with a protein. So there's per one protein, there's one binding pocket. And from this siderocalin, we can then engineer something like this, where we have this antibody that knows where to go inside the body, it knows where to target, and then we can just attach two of our proteins. So this trastuzumab or TMAB is a well-established targeting vector. People have done a lot of studies, years and years and years of studies that this can target. So what's novel here, what's new, is using this fusion protein idea to attach these siderocalin proteins. So our collaborators have established that, here I'm just showing some classic characterization of these proteins, and that they did some immunofluorescence staining showing that these proteins can uptake in cells. So how does this relate to a theranostic? I'm getting there. Theranostics, if we want to try to do something with imaging and therapy, I've talked a lot about zirconium-89. Zirconium-89 is a pretty interesting positron emitter because it's actually a 4+ positron emitter. And so what's the analog for that, or vice versa? If you're thinking about thorium-227, what's the imaging analog of that? So thorium-227 is an alpha emitter. And these two have been used before as a theranostic pair. Zirconium for imaging, thorium-227 for therapy. What's really interesting about thorium is that actually as it decays, it emits 4 alpha particles. Alpha particle radiation actually has a shorter range in tissue. So if you want to do a beta particle, like what I showed before, the lutetium-177 that was FDA approved, those travel much further. And so the problem with that is that if you have small tumors, then you might be damaging tissue that's actually healthy tissue. So an advantage for these alpha particles is that you get it directly to the tumor site, you have no off-target tissue uptake. So based on trying to design something using thorium and zirconium, we've already established that these HOPO complexes and CAM complexes are recognized by the protein. Basically, that means that we have to pick the correct chelator to put inside our protein for whichever metal that we're using. And since we know that zirconium is bound by CAM and thorium and that they have high affinity, that's the chelator we're going to go with. So we actually went with a mixed chelator that has some of both of these properties, and I don't need to go into it right now, but the point is that this protein has specific affinity for different charges of metals. And so depending on which metal you're using, you have to think about which chelator you're using. So we're going to use this mixed ligand. We're going to swap out either zirconium for imaging and thorium for therapy. So first we're going to label it, and then we're going to put it into our protein. And this is our hypothesis, but this will just be a lot simpler. Just put it into the protein and then put it in vivo. You have to way less synthesis of trying to design a bioconjugate complicated chelator. So I want to talk briefly about the workflow. I've mentioned theranostics and chelators and synthesis, but I kind of want to give you context of what we do day to day to validate these things and how they actually go from just ideas and rotovacting things to putting it into preclinic and putting it into clinical trial. First, you have to synthesize your chelator. This chelator is the one that that colleague of mine synthesized, and it's that ligand that I showed you before, multiple steps, but it's not as complicated as trying to develop a bifunctional chelator. So first you synthesize it, and then you characterize it. You want to characterize it with metals that are cold. You don't necessarily have to jump right into radioactive work. You want to just validate that it binds what you want it to bind. And then you perform all of the radiolabeling studies. After you validate that it actually binds the cold metal, you want to validate that it binds the hot metal, the radioactive metal. Then, before going into mice, you want to actually validate that it targets the cell. So you'll put it into the protein, and then you'll actually do in vivo cell before you ever even go into the mouse. Then, ideally, you go in and you image, and you detect via PET imaging where these tumors are, validate via looking at the tumor uptake that there is uptake. And then following the imaging validation, you'll perform therapy studies and hopefully see, I mean, this is cartoonistic, but hopefully you see just complete retraction of all tumor tissue. So first we did with the fusion protein, we did exactly this process. We put the radiolabeled and put the complex into the protein. We validated serum stability over time. So what that means is we're not putting it into the mouse yet. We're literally just taking our protein complex. We're putting it into an Eppendorf tube that's filled with serum. It's basically just, like it's a plasma mimic. And so, or it is plasma. And then we heat it at 37 degrees, which is your body temperature, over however long. And we just look at, is it falling apart? If it falls apart, don't need to put it in the mice. That's, that's not, waste of mice. So it looks like it's doing okay. Right now, the thorium, it looks like over time, right? % intact complex over a period of time. Maybe the thorium isn't doing so well, but that could be because of the alpha damage. We don't actually know yet. We then validated in cells. Basically, just take your complex, you grow your cells, incubate with the protein, and you can observe uptake. We're basically comparing. This is the targeting TMAB. It uptakes in the cells. This is the non-targeting siderocalin. No TMAB, and not as much uptake. And that's simply it. That's all it really takes. Yeah.
How long do you expect the compound to circulate, like in the body before it gets treated, like where on that time scale?
Yeah, that's a great question. For these large antibodies, it's a large antibody and it's large proteins attached to it. The larger you get, the longer it's going to take to excrete from the body. So if you have a smaller peptide or a smaller protein, it's going to circulate more quickly in a few days, probably. But these peptides will probably circulate for several weeks. Yeah. So up next is exactly visualizing the circulation, trying to understand where is this protein going and is it performing uptake in the tumor. So the experiment is to formulate the protein, inject, and then image at different time points for biodistribution. So here what I'm showing are PET images of our patient, our mouse, and this is the non-targeting. So I have two different views for you. Coronal is on the left, so coronal, and that's again where the mouse is like this. So you're looking at head up here and tail down here, and then sagittal is mouse on the side. And so the arrow is pointing to the tumor, and you're like, well, what am I looking at? I don't see anything. And that's because this is the non-targeting construct. So this is good. We would be a little concerned if we formed a non-targeting construct and it started uptaking in the tumor. That would be confusing. Here is the TMAB. So it's our whole construct that targets, and you can see actually really nicely over time, it has prolonged uptake into the tumor right here. And you can see it on the side as well. And so this is our visualization of uptake in the tumor. And this is what we would want to see. We're really, really excited for this. Yes, it's targeting. So we can also do biodistribution. That basically gives us, again, the information of how much of the zirconium is in which organ. And so we look at all of these different organs, and there's a couple different controls up there. This is after 120 hours. The main thing I want to show you here that you can focus on is here again, tumor uptake. With the targeting, we see much, much higher accumulation into the tumor compared to a non-targeting control. And we also see excretion through kidneys and spleen, which indicates to us that it's not going to the bone, it's not falling apart. The zirconium isn't just being ejected from the chelator and going straight to your bones. It's actually staying securely within the protein and just getting excreted, which is great because we both want to see tumor uptake and that this thing is not falling apart. So now that we've established that we can see it, it targets those cells, we can see uptake. Next is alpha therapy. So easy peasy, just switch it out with thorium, right? There's actually a lot that goes behind this. We actually have to purify the thorium, and we have to figure out the radiolabeling conditions. It's a very different metal. But in simple practice, we just inject, and then we literally measure the tumor size over a period of time. And so what I'm going to show here, we're still collecting this data as of today or tomorrow. So here, what we're looking at is the tumor volume as a function of days elapsed. And so here, this is each line represents one mouse. Here you can see some tumor growth. This is our saline control. Here's the non-targeting. So you can see some growth here, which is not great, but it's also not non-targeting. And then we see then some decrease here of the tumor volume with the targeting construct. So this is data that we're still processing and piecing together and analyzing, but what this is showing us is that if you switch out zirconium for thorium, depending on how much dose that you're giving, that's one thing that we're working on. We could have given them a little bit more thorium than we did. Then you can see a tumor treatment or tumor regression. So all this is to say, imaging and therapy together, we think that this, we can establish that this fusion protein does target and perform a therapeutic effect inside this mouse model.
I'm sorry, just the units, tumor volume is cubic, is that cubic centimeters? That's huge, isn't it?
Yeah, these are better. These are, this is almost the end of the study. Yeah, this
Okay.
is pretty big.
Wow. Yeah, so it's like 10 centimeters by 10 centimeters by 10 centimeters. It must be millimeters, it must be millimeters.
Yeah.
It has to be millimeters, right, I was gonna say that it's maybe the
Oh,
mouse.
no, yeah, you're right.
Okay,
It's funny, we looked at this
Okay,
data
yeah.
for so long, we haven't caught
Yeah.
that, so thank
yeah,
you.
that's cool, that's cool.
It's cubic millimeters, not cubic
Blobby mouse, but that's fine.
It's tumor with
Well,
side
I
of
call
mouse.
it.
1,000 cubic centimeters.
Yeah.
Is a liter.
Is each line like a
1,000 cubic
individual
centimeters.
mouse?
Yeah, this isn't necessarily how we would publish the data. I'm just showing you the raw data for now.
Right,
Yeah.
might be a dumb question, but how did you assess the volume of the tumor without removing it from the mouse?
The tumor xenograft, the way this works is we're not guessing where the tumor is. We put it in a very specific location and we watch it grow and we monitor it. And so it not centimeters, millimeters. We literally just use a caliper and go in. Alex actually goes in very frequently and measures just the width and height and we calculate the volume. So that's the advantage of these studies is we don't have to sacrifice them to watch the tumor treatment. Yeah.
So xenograft means you actually grafted it on,
Yes.
you gave this thing.
Yes.
Cool.
Yes.
And each line is a mouse, is that the idea?
Yes. Yes.
Oh.
Yeah. So I think just to summarize, I wanted to bring back. I've talked a lot about half-lives, and I think that's a great way to kind of contextualize the different types of in vivo work that we do. And again, just kind of coming back to, it really comes back to the radioactive fundamentals. Talked a lot about coordination chemistry. That's my home, so happy to talk more about that. But ultimately, trying to use chemistry design, we can focus on improving the therapeutic effect from our different theranostic agents, which can lead towards and has, we have these radiotheranostics that are ready and in FDA approval and stock image of global treatment. The last little tiny bit that I just wanted to really quickly touch on is if you know me, I will be talking about Cherenkov radiation because that's just my passion and my heart. So Cherenkov radiation is just this really interesting phenomenon where when radioactive particles at a certain energy threshold are released in a dielectric media, they emit this blue glow. It's kind of a sonar wave of energy. And this blue glow is actually, this is all out of order, is actually, you can observe it as the photons emitted as a function of wavelength. It's this blue shifted energy. And so you can do really interesting things with it, like trying to throw in zirconium and excite lanthanide complexes, which works, and I think is really interesting. But I think there's a lot of potential to try to understand actually alpha radiation damage using interesting isotopes such as actinium-225, which have multiple daughters that decay and have multiple interesting properties. So actinium-225, it's an alpha emitter, so we wouldn't necessarily be understanding, it's not going to generate Cherenkov emission on its own, but the daughters such as bismuth, francium would have bismuth would have Cherenkov emission. So I just wanted to touch on it. It's going to be really interesting moving forward. So anyway, all of this work, not just me, huge team effort. I see my group members here. Jen, all of you guys have done so much work in bringing all of this together. So I want to thank you all. It's a huge team effort. So I'm really proud of my group. And thank you, Rebecca, for letting me present all this work. Well, take it away, bye.
The targeting molecule is the molecule that has two proteins on it that are fused together, right? With a ligand. Okay, so one of those is the targeting molecule, the other one is the therapeutic model
Yeah, let's see if I can go
molecule.
just right here. I have it everywhere.
Right.
This right here, I'm cartoonizing it, but this is an antibody and it's the trastuzumab antibody. And so this antibody is well established to target the HER2 receptors of both ovarian cancer
I see.
and breast cancer. So we didn't want to, not too many variables, right? We're just establishing that this targets with those additional
So
proteins.
those are two identical molecules
Yes.
that are connected to the one antibody.
Yes, which is actually also, I didn't highlight this, but this is a huge advantage of this system. It's very difficult to actually get one targeting molecule with one radioisotope. So the fact that we're getting double bang for our buck, more alpha emitters targeted, so that's an advantage as well.
More questions?
What kind of answer? But how far away is HOPO from its FDA approval?
Ooh, close. It's in process right now. I don't know all of the most recent updates, so I won't say anything. But it's on its way, yeah.
Any other questions? Let's take Alexia.